Good morning, everyone. Now I'll discuss briefly about our laboratory diagnosis of SARS-CoV-2 or the COVID-19. And this discussion, it has been divided into two parts. One is molecular testing, which I will discuss. Then Dr. Sangram Singh Patel, he will discuss about the serological testing. So this you have already seen. This just to show that what are the structure of this virus and what are the various targets. Like this envelope is common in all beta coronaviruses, right? So it does not tell you that whether it is SARS-CoV-2, but it will be positive in any coronavirus. So it is used as a target in screening test. Then we have other targets like spike, glycoprotein, and hemagglutinin, esterase dimers, and others. So there are various tests, but molecular testing, it is the key or the core for the diagnosis of SARS-CoV-2. So like if you wish to have a good plant, you need to have a very good seed. Similarly, if the test has to be good, we have to have a very good sample. It is dependent on the sample. So how the sample has been collected and how it has been transported and how it reaches us, that becomes a very important, and we are dependent on that. Only the testing methods, of course testing methods has to be foolproof, that we do, but it is largely dependent on many factors. So that is very important to understand, and therefore sample collection and transportation forms the very basic for the lab diagnosis. And real-time PCR, that is RT-PCR, is the major workhorse in the field of molecular diagnosis. It can amplify and detect, theoretically speaking, a single copy of a specific genome sequence, and therefore it is extremely sensitive. And as you have seen, it is a RNA virus. So therefore we have to do RT-PCR. It is a quantitative technique, and RNA copies increase exponentially, and they are proportional to the amount of starting material, that is the initial viral load. And various targets, as I've told you, that is an envelope or nucleocapsid or spike or RNA dependent RNA polymerase and ORF gene in various like kits or that we get, and it is said that at least usually 2 to 3, depending on the kit, they target on, either they detect 2 target or 3 targets, but minimum 2 is preferred. So this is in brief about the method. That you have to collect the nasopharyngeal swab by the proper method, and then the specimen is stored. It is better to be transported immediately at 4 degrees centigrade, or in the ice. We have vaccine carriers and other methods for that. And if it is being collected in the district, then it has to be transported as soon as possible, at the most like maximum up to 72 hours, but earlier the better. Then RNA is extracted, and then you are seeing such we have such type of machine called PCR machine, PCR machines, where the amplification is done, and then we get such type of graph. So estimated time for a single, this in the whole process, this is a lengthy process, and so it takes about 10 to 12 hours, including the extraction process. Then there's something called CT values. It is the number of the PCR cycles which are needed for a detectable fluorescence, that is the greater than the fluorescence signal threshold. As you can see, there is a baseline or the threshold, and here this is the threshold, and then we see this rise. So in various kits, the CT value is different, but usually it varies from 35 to 40. So in kits, it is somewhere it is 35, somewhere it is 40, at which it is taken as positive or negative. So there are various type of kits. In some, it is you have to do 2 assays, like one is screening assay and another is confirmatory assay. But now we have multiplexing also, that you have to do a single test and it will give you the confirmed diagnosis. Now this is very important. This is showing you need here you can see that PCR, this RT-PCR, it becomes positive much before the patient develops some symptoms. That is even before 2 to 3 days before the symptoms. Then it rises, it reaches its peak at around the first week at around say 6 to 8 days or 9 days, and then in the after the second week, it gradually declines, and after the third week, it slowly decreases and then gradually becomes undetectable. And regarding the serologic other testing, its correlation with other testing like antigen detection test, antibody, all this Dr. Sangram will tell you. But briefly, this antigen detection test, it is you have to do this test. It has to be done within the first week. That is in first 6 days of illness, as you can see here. And after that, it declines rapidly, and this antibody detection test only after the second week of test, and it is usually not for the diagnosis. So at what time the patient, time of presentation, at what time this is, and that becomes very important, and accordingly you have to choose the test. So a positive PCR results reflects only the detection. It is important to understand that it reflects only the detection of viral RNA and does not necessarily indicate presence of viable virus, because it will detect both live and dead. And in some cases, as I told you, it usually after 2 weeks it decreases. But in some cases, RNA are detected beyond 6 weeks following the first positive test. So varies from case to case. And in literature, few cases have been reported positive after 2 consecutive negative PCR tests when they are performed 24-hour apart. And it is not very clear whether it is a testing error or is a reinfection or reactivation. There are false positive and false positive results as in any test. So false negative results may be due to improper sample collection, type of the sample, techniques and the timing, or there may be degradation of the virus during the shipping or the storage. Then there may be presence of RNA RT-PCR inhibitors in the sample, or if the virus has mutated and our target is different. So naturally, with the existing method, it won't be able to detect the mutated virus. Then each test kit has different sensitivity and specificity, so it depends on that also. False positive results may also arise due to cross-contamination during specimen handling or preparation, and RNA contamination during the product handling. So false positive may also occur, and interpretation is very important. Then we have a TrueNAT test, which is an assay for screening and confirmation of COVID-19. It is a chip-based RT-PCR, semi-quantitative, and it detects beta coronaviruses. It is very heartening to note that it is manufactured by India, and samples are again oropharyngeal swabs are taken, and various targets are mentioned here. Here the sample is collected in a viral lysis buffer, which is a bit different from viral transport media used in RT-PCR. And now we have 2 tests, like step 1, which is screening method, and if it is negative, there are 2 tests currently we have to do. E-gene we detect in step 1, which is a screening assay, and then if it comes positive, then we have to go for confirmatory assay. But recently a multiplex assay has also come, where it can be done in a single step. And this is our data. Though the manufacturer reported 100% sensitivity specificity in a small sample size, but we have around 1200 samples. We have done both the tests, and in our hands the sensitivity is around 70%, specificity is around 88%, and its negative predictive value is very high, that is to the tune of 95%. So it is a good test. Okay, sir. Then so this was about the basic test. Now there are certain point of care tests, right? Point of care tests means this can be done at the patient's or the bedside. So there are few, like this one, the Cepheid one. This is it takes around 40 minutes, 45 minutes time. Principle is also the same, and they have different targets. This I forgot to tell you something about the TrueNAT and cartridge-based test. There are some differences. Like TrueNAT, it can be done in simple settings, villages, because it is battery operated, doesn't need power supply. AC is not required. It is very cost effective, indigenous, and less risk because it's a closed system. And as compared to the cartridge-based system, then we have another 2-3 other point of care test. But the problem with this, which can be done in a very short time, like sample preparation time 5 to 10 minutes, then in 10 to 15 minutes reporting. But the problem is that they are all of them, they are very low throughput. So when you have to do a very few tests, these are useful. But in a large setting like us, these are not, neither they are cost effective nor they are very useful for settings like us. or in a country like India. So there are various type of tests are available in the market, but the principle remains the same. So I'll not go into the detail. Then we have this this BioFire. There, apart from this, you can detect other viruses also. So thank you very much.